I'm very happy to present our next speaker, Dr. Vicki Hertzberg. Dr. Hertzberg is a professor at Emory's Nell Hodgson Woodruff School of Nursing. She is an internationally recognized expert on big data and its impact on healthcare, and is widely known for her work measuring the social contacts in emergency departments and disease transmission on airplanes. Her research has been funded by the National Library of Medicine, National Institute of Neurological Disorders and Stroke, the National Institute of Environmental Health, and the National Institute of Allergy and Infectious Disease. Dr. Hertzberg is the founder and director of Emory School of Nursing's Center for Data Science, and she also directs the Data Science Core of the P30 Center for Symptom Science, Metabolomics, and Multiple Chronic Conditions, which is hosting this conference. Today, she'll be speaking to us on multi-omics and the progression of ALS. Please join me in welcoming Dr. Vicki Hertzberg.
Good afternoon. My name is Vicki Hertzberg, and I want to talk to you today about using multi-omics to study the progression of amyotrophic lateral sclerosis. Amyotrophic lateral sclerosis, also known as ALS, and sometimes commonly known as Lou Gehrig's disease, happens when the motor neurons, the neurons that travel from the brain down to the muscles, die, and therefore then you can no longer stimulate the muscles to contract or discontract, and there's no known reason why this happens, and there's also no cure. There are a few FDA-approved drugs that will delay the progression of the disease, but they will not cure it. So we wanted to look around and see what the evidence was as far as how does the gut microbiome have any part in ALS. And so we endeavored to do a little pilot study. And there was at the time only one study in the literature in mice that showed anything, that there seemed to be something going on in ALS mouse model. So we went to do this study, and for the controls, we chose the partner or caregivers of the patients. They were all spouses, so now I'll call them spouses. And we consented them in clinic, and there we obtained two different types of samples. We asked the patients and their spouses to take rectal swabs, and from these rectal swabs, we obtained samples of stool. And we also did a blood draw for venous blood to convert into plasma. Now we took the stool and the plasma and we did the following assays. First of all, we sequenced the 16S ribosomal RNA gene from DNA isolated from the stool samples to determine the microbial populations that were present in the patients and their spouses. We also, from both stool and plasma, determined levels of inflammatory biomarkers. And then finally, of the plasma, we did metabolomic analysis, which was an untargeted analysis. So in terms of demographics and clinical variables, our patients and spouses were roughly comparable in age. Because they were spouses, they ended up being diametrically opposite in terms of gender, and they were almost all white. For the patients, their disease duration in years was less than 2 years. And this was actually a study entry criterion. We tried to get patients that were within 2 years of disease onset. No patients were taking riluzole, which is one of the FDA approved drugs, and it was the only thing approved at the time. No patients had a family history of disease. No patients had the C9orf72 mutation, nor did they have the SOD1 gene mutation. These mutations have been associated with ALS and other studies. And no participants had history of other chronic diseases. So what did we find? Well, in 2020, we published the results of our 16S gene sequence evaluations. And here we found that the ALS patients' gut microbial populations were actually more diverse than the controls, which was a surprising finding for us. And this was initially done in a blinded manner, so all we knew was that the two groups were different. And we guessed that the ALS patients were less diverse, and it turns out we were wrong. So Chow-1 diversity measure indicates it's roughly equivalent to the number of different types of microbes that are in a population. And then the Shannon diversity talks about the evenness, I mean, how well they split into the different subpopulations of different bacteria. And in both of these measures, the ALS patients were higher than their spouse controls. We next did a principal coordinate analysis that showed that the patients